And I'd like to invite the third and the last speaker for today. The last speaker for today's session is from the Northwestern University of the USA, and he's currently working for the Feinberg School of Medicine. We have Professor Judd Hultquist. Professor Hultquist, I would like to introduce about his bio. He graduated from the Market University, and he got the BS for the Market Chemistry. He graduated from the Minnesota University, and he got the PhD with the Molecular Biological Studies. And he studied the biology. And in San Francisco, in the David Gladstone Institute, he finished his postdoc course. In his lab, he is studying lots of the infectious disease viruses like Ebola virus, HIV virus, and even the SARS virus, and even the MERS virus. And he is studying lots about the virus to the human cell interactions from the system's biological perspectives. He recently studied a lot about the coronavirus, and then his articles were published in the Nature and other renowned journals. Today, his topic is SARS-CoV-2 mutation. In the keynote speech, we heard about the mutation, and this mutation research will be the foundation and critical for the development of the treatment and for the development of the vaccine. He studied the mutation of the SARS-CoV-2 mutation, causes, consequences, and the opportunities. That is the title. And he's going to give us the explanation based on the U.S. cases. And I would like to invite Dr. Hultquist.
Good morning, everyone, and greetings from Chicago. I truly appreciate this opportunity to share our recent SARS-CoV-2 work with you all, and would again like to thank the organizers for their very kind invitation. So today, I wanted to talk with everyone about SARS-CoV-2 viral diversity, causes, consequences, and opportunities. This topic was inspired by all of the recent stories in the media about SARS-CoV-2 mutations and their potential impact on the pandemic. While the idea of a virus mutating may seem scary, most mutations are in fact harmless and serve as a remarkable tool for us as researchers to track viral spread and inform public health policy. So let's take a bit of a deeper dive on the causes and consequences of SARS-CoV-2 viral diversity and look at some of the opportunities that this diversity affords us. The first thing to understand is that all viruses mutate or change over time. These mutations can be sorted into two basic classes: intrinsic mutations and extrinsic mutations. Intrinsic mutations arise due to internal properties of the virus, such as the fidelity of the polymerase or the ability of the virus to proofread its genome and correct its own mistakes made during replication. Extrinsic mutations, on the other hand, arise due to the influence of the cellular environment, including imbalances in the concentration of nucleic acid building blocks and the presence of DNA or RNA editing enzymes, such as the ADAR or APOBEC family of proteins, respectively. Both of these kinds of mutations together define the viral mutation rate. The second thing to understand is that different viruses mutate at different rates, as shown in this chart here on the left, comparing genome size to mutation rate. Generally, the longer the genome, the lower the mutation rate. This truth holds true from eukaryotes like us with very long DNA genomes, through prokaryotes or bacteria, and viruses. RNA viruses in particular have some of the highest known mutation rates recorded. HIV and influenza viruses, for example, have notoriously high mutation rates that have resulted in significant problems in vaccine design and antiviral resistance. Just as an example, HIV makes an error in 1 of every 5 genomes produced on average. In an infected person, not on antiretroviral therapy, as many as 10 billion to 1 trillion new viruses can be made each day. That means the virus can explore an incredible amount of mutational space even within a single individual in a single day. Now, if these mutations fall in drug or antibody binding sites, they may result in antiviral resistance mutations that render treatments ineffective. This is illustrated by the cartoon on the right. If we have an initial infection event, indicated on the left of that diagram, the virus can diversify within a host over time. Now, if a mutation is present that offers antiviral resistance, that population can grow out even in the presence of drug, and the disease will persist. Of course, if this selective pressure is removed, the virus can diversify once again. Besides contributing to antiviral resistance and immune escape, some viral mutations have also been observed to influence the spread or transmission of viruses, both in moving between species and in moving between individuals. For example, HIV acquired a number of mutations to effectively jump from monkeys into the human population. These mutations were required to overcome the human immune response and optimize the virus for replication in a new species. Similarly, during the 2014-2016 Ebola virus outbreak, some mutations arose that were found to improve human-to-human transmission of the virus. Now, coronaviruses, like SARS-CoV-2, also required mutations to jump into the human population. The original SARS coronavirus that caused an outbreak in 2003 was thought to have come from bats through civets. MERS coronavirus, on the other hand, which caused several outbreaks in the Middle East in 2012, was thought to jump into humans from camels. With SARS-CoV-2, there is some evidence that it may have come from pangolins, but that still remains under investigation. In all of these cases, viral mutations were acquired to facilitate replication in the human population. And, of course, the virus is continuing to mutate within humans. Researchers from across the globe have been sequencing the virus over the course of the outbreak and have been finding a vast array of viral mutations that have been arising in different places at different times. Luckily for us, SARS-CoV-2 has a much lower mutation rate than HIV or influenza. If we refer to the same chart we were looking at earlier, it falls on the extreme low end in terms of mutation rate for RNA viruses. Now, this is primarily due to two things. First, the SARS-CoV-2 genome, shown in the upper right, is huge for RNA viruses. It is among the longest genomes of any known human RNA virus. As such, the polymerase is thought to be more accurate out of necessity. The second thing to note is that the virus encodes its own proofreading enzyme, known as NSP14, that helps to correct errors and lower the mutation rate even further. So yes, SARS-CoV-2 is mutating, albeit at a lower rate than most other RNA viruses. So the question now is, 1, how can we detect which mutations might be harmful? And 2, how might we use these mutations to our advantage? As it turns out, there's a lot you can learn from a few mutations. While some mutations may be harmful as they provide antiviral resistance or improve transmission, these are the exception to the rule. A vast majority of mutations are neutral or even deleterious to the virus, and they can be very helpful in informing researchers as to how, where, and when a pathogen might be spreading. For example, based on how similar two virus genomes are, we can infer their ancestry, as shown on the far left. Indeed, given the somewhat predictable mutation rate of a virus, the number of mutations can be used as a sort of molecular clock to estimate when certain mutations arose. The critical mutations at each step can be flagged for functional and structural analysis to predict which ones might cause antiviral resistance. Finally, if spatial metadata is associated with these samples, the routes of transmission can be inferred from the trees, as shown on the right. This type of research is being done around the world to inform vaccine strategy, public health practices, and clinical protocols. So to give you a real-life example, I now want to do a deeper dive on what we're learning here about the pandemic in Chicago, Illinois, in the United States. Just so we're all on the same page, I wanted to give a brief rundown of the pandemic in the US. In this chart, case count each day is shown by the blue bars with a rolling average indicated in the orange line. The first confirmed case in the United States was reported in Washington State on January 20th, followed very closely thereafter by another case on January 23rd in Illinois. Now, the first confirmed case of a person-to-person transmission event was recorded in Illinois shortly thereafter. The virus wasn't reported on the east coast of the country until March 1st, when the first confirmed case in New York State was reported. Now, shortly thereafter, infection rates start to rise, and around the country we saw stay-at-home orders put in place, with Illinois, New York, and Washington State all issuing these orders around March 21st to March 23rd. Shortly after the stay-at-home orders went into place, we quickly hit several sad landmarks in the epidemic, including 1,000 deaths on March 26th, 10,000 deaths on April 6th, and 100,000 deaths on May 28th. Despite the continuing case numbers and continuing deaths, all U.S. states began reopening phases in late May. That led to a rapiding of cases that we have called the surge, where we've seen 2 million cases by June 11th, 3 million cases shortly thereafter on July 7th, and 4 million cases on July 23rd. In fact, we've just recently hit 5 million cases. So what I want to talk about today is to focus in on the very early events of this pandemic during March and April. But the same techniques that I'm showing here are going to be applied to the rest of the pandemic to understand these later dynamics, particularly those that have to do with the surge. Now, that was looking at data in the United States as a whole, but we also have more granular data looking at each of the 50 states. And when we separate out the total number of positive tests as a proportion of the population in each of the different states, we see quite different trends over time. So, for example, in Illinois, shown here by the black line, despite seeing early introduction of the virus in January, we didn't really see significant rise of cases until sometime in early to mid-March. Despite that rapid rise, we still leveled out at a relatively low case count. Now compare that to Washington State, on the other hand, which saw a similar time of introduction in January, but saw a very rapid rise in case counts. Despite that rapid rise, it also petered out at a relatively low total. Now, New York State, which saw a relatively late introduction of the virus, saw very rapid increases in case count and higher numbers overall. Now, this can be due to a number of different factors, including differences in population density, demographics, community mitigation measures, differences in testing, differences in healthcare infrastructure, differences in public health reporting practices. But what we were interested in is whether or not this could also be due to differences in the sequence of the virus that was impacting the different U.S. states. In other words, are there differences in viral mutation that could account for the differences in these trends? So to address this question, we collaborated with the hospital here at Northwestern, and we were able to obtain 127 nasopharyngeal or bronchoalveolar lavage swabs from mid to late March. From these samples, we extracted RNA, reverse transcribed it into complementary DNA, and then amplified the SARS-CoV-2 genomes by multiplexed PCR. These PCR reactions were then barcoded and subject to deep sequencing on the nanopore platform. We were then able to assemble the whole viral genomes of 88 of these clinical isolates. What we found was actually quite interesting. So all of these samples were taken at the very beginning of the epidemic in mid-March, all from a single hospital. But despite all of the samples being taken at the same point in time in the same hospital, we saw quite a bit of genetic diversity. In fact, we saw 3 distinct clades of virus, or families of virus, coming up within this single patient population. This first and largest clade we're calling clade 1, the second clade 2, and a small third clade. For those phylogenetic experts in the audience, I just wanted to let you know that clade 1 corresponds to the pangolin labeling system of B1, clade 2 corresponds to A3, and clade 3 corresponds to A1. So to better understand where this sort of viral diversity came from, we wanted to better take a look at how these sequences corresponded to other sequences in the United States as a whole. So we downloaded all available SARS-CoV-2 genomes available in the United States at the same time point in mid-March, and did a phylogenetic analysis of our sequences relative to the rest of those in the United States. The sequences that we sequenced here in Chicago are highlighted by the green large circles. And what we saw was that the genetic diversity in Chicago was broadly representative of the genetic diversity we were seeing in the United States as a whole, with clade 1, clade 2, and clade 3 being represented. What was interesting was that clade 1 was most consistent with the same viral strains that were circulating in New York State on the East Coast, while clade 3 was most consistent with strains circulating on the West Coast in Washington State. However, these clade 2 viruses seem to be relatively unique to the Chicago area. Now we can map these geographically to better understand where these transmissions were coming from. And again, as we saw with our phylogenetic tree, it appeared as though the clade 1 viruses originated in the New York area and rapidly spread around the United States. The clade 3 viruses, on the other hand, originated in Washington State and then spread primarily to the West Coast and Midwest, while the clade 2 viruses were almost entirely found within Illinois. So to better understand the sequences in the context of the global pandemic, we did a similar analysis. We downloaded all available sequences globally from the same time point in mid-March. And what we found was that while Chicago's genetic diversity was broadly representative of the global pandemic, there were several clades that were not found in Chicago that were found elsewhere in the world. Nevertheless, we did see similarities between distinct regions around the globe. For example, the clade 1 viruses, that were very similar to those we found in New York, were also highly prevalent in the European area, while the clade 3 viruses and clade 2 viruses, representing the Washington State and Chicago clades respectively, were most similar to those sequences found early in the epidemic in China. So to better understand where the Chicago-specific clade originated, we performed a phylogenetic analysis of this clade in particular. And what we found was quite interesting, in that the sequences were most related to those sequences originally identified in Wuhan and subsequently and elsewhere in China. So what we believe happened is that a visitor to China returned to Illinois and initiated community spread sometime in early to late January. That spread continued until we sequenced the population sometime in mid-March. We detected very rare transmission events outside of Chicago after that time point, with the exception of another cluster being detected in Australia. We believe that that cluster originated again from a transmission event from Illinois to Australia. So the local SARS-CoV-2 epidemic in Chicago is broadly representative of that in the US and of the globe itself. Just to recap, we hypothesized that the Chicago-specific clade 2 viruses originated directly from China and came to our city early in the pandemic. Clade 1 viruses, on the other hand, came from the east by way of New York and Europe. Clade 3 viruses came from the west by way of Washington State. Relative to the other two clades, clade 2 viruses have spread relatively little, while the clade 1 viruses have expanded rapidly, not only across the United States, but across the globe. So the question now is there a specific viral mutation responsible for the limited spread of clade 2 versus these clade 1 viruses? So to better understand that, we compared our viral genotype data with those characteristics and outcomes of the patients from which the isolates were originally obtained. We were actually kind of fortunate in that Chicago was a melting pot of virus diversity, and we could directly compare the clinical and demographic characteristics associated with each viral strain in a single hospital system. This is very important as it controls for a lot of confounders regarding diagnostics, standard of care, and missing values that can otherwise plague large clinical data sets compiled across several hospital systems. So to do this analysis, we fit a linear mixed effects model to these data to account for covariance. When we did this, we found no significant association between our various viral clades, that is, our various viral mutations, and any demographic or clinical variable, including sex, age, BMI, race, ethnicity, disease severity, isolation source, or date. However, we did find a significant association between the clade of the virus and the CT value, which is a measure of viral load. That data is illustrated with these box plots shown here. We're looking at CT value, again, which is a measure of viral load, shown on the y-axis, and the various clades depicted on the x. And what we found was that clade 1 viruses had significantly lower CT values, meaning they had significantly higher viral loads compared to viruses in these other families. Now, it's important to understand whether or not this is due to the fact that these patients truly had more viruses in their nasal passages at the time of diagnosis, or if there were differences in the time that the sample was taken. What we've found by studying hospitalized patients within Northwestern was that over the course of infection, viral load tends to drop regardless of clinical outcome. So it was important to understand if the time at which the samples were taken was consistent between our different viral clades. When we did this analysis, comparing either time to symptom onset, shown on the left, or time to possible exposure, shown on the right, we saw no significant differences between our clades, indicating that the most likely explanation for these differences is that the patients had higher levels of virus if they carried a clade 1 virus compared to clade 2. Also importantly, we found that viral clade did not appear to be associated with disease severity, but again, more study will be required to take a look at this. So overall, we hypothesized that clade 1 type viruses carry a mutation that enhances viral transmission. And this has been borne out by our more recent sequencing efforts. So all of the sequences I had been talking about earlier all originated in mid to late March, but we have since sequenced more viral isolates from patients at our hospital in April and in May. When we did this, we found that this clade 1 really became predominant in our area, suggesting that it may have improved fitness relative to these other viral clades. So to determine which mutation might be responsible for that, we were able to look back at the viral sequences and determine which mutations were critical for defining those viral families. There were several mutations that we found, but the one that stuck out to us the most was a mutation in the spike protein, D614G. Now, we're not the only people to have seen this mutation in our sequencing. In fact, a really excellent recent paper looked at the prevalence of this mutation across the globe from January through May. And they found, similar to what we found, that over time, the G variant of this mutation became more and more prevalent across the globe. In fact, when they engineered this mutation into new viruses and infected cells ex vivo, that is, in cell culture, they found that viruses containing the D614G mutation, shown here in blue, had higher viral outputs than the D614 variant. So the big question is, does D614G alter viral transmissibility? We know that viruses carrying that mutation have higher viral loads in their upper airways. We know that viruses with the mutation have become predominant across the globe over time. And we know that viruses with that mutation infect cells more readily in vitro. But before we can definitively make the statement, we need a good transmission model to determine the impact on transmissibility, and a good one has not yet been described, though there are many efforts to develop these transmission models in either golden hamsters, mice, ferrets, or even monkeys. So there are multiple other mutations that our analyses also discovered, including some in clade 3 that seem to inhibit essential viral proteins. And by doing structural analyses, we're identifying mutations that may contribute to antiviral resistance in the future. So to summarize, all viruses mutate and change over time. Most of these mutations, however, are harmless. But in rare circumstances, they may influence viral pathogenicity. Luckily, we've developed the tools and techniques to use viral variation to monitor disease epidemiology, and this can be an incredibly powerful tool to informing public health strategy. Now, SARS-CoV-2 may have evolved to transmit better in the human population, but more study will be required in animal models yet to be developed. And with that, I would like to thank my incredible team of collaborators here at Northwestern who put in just an extraordinary amount of effort to make sure these studies got done. I'd also like to thank our funding sources who made this work possible. And finally, I would like to direct everyone to our preprint publication available now on medRxiv. Thank you all again for this wonderful opportunity, and I hope you enjoy the rest of your conference. Thank you.
That was an easy explanation and detailed explanation. Thank you very much. And that was a really wonderful lecture. I would like to thank Professor Hultquist once again. He talked about the virus mutation definition, and he gave detailed explanation about the virus mutation. His case was centered on the Illinois, Chicago, and he said the virus clade and the transmission rate can be different. And if we understand the clade and the transmission rate, and probably this knowledge can contribute a lot to the development of the treatment and the therapeutic mechanisms. And also, we need to understand the reason for the mutation, and he gave us a full explanation about the characteristics for each of the region. Just as in the contents of the keynote speech, the D614, DNG, so that kind of mutation can increase the transmissibility. And after listening to these three speakers, we were able to understand the basic science surrounding the COVID-19. And also, I would like to thank all the speakers who gave us the video lecture. Dr. Shin, Dr. Chang, and Dr. Hultquist, thank you very much for the wonderful preparation. As Dr. Chang and the Hultquist said, it's going to take some time for us to come up with the treatment. However, they are making lots of contribution to the scientific discovery and development. And I am sure we have a promising future to overcome the coronavirus. And please encourage them so that they can study more about the viruses. I sincerely hope that we contributed a little bit more for you to understand more about the COVID-19. Thank you very much for staying with us for a long time, especially those who are online. Thank you very much for your participation. And this concludes the session number 1. Thank you.